Oh, all right. We're going to start off and start the review on chapter 3. All right. Again, now this, I really wanted, this is one of the reasons why I wanted to record this part, is because chapter 3, I did not make the PowerPoints, and I'm not going to have a chance to go back and make them before the test. So if I do, if I do get a chance to, I will do my best, but more than likely I won't be able to. So chapter 3 is all about the cell. And so we were looking at this is how uh, we had that build up. We had atoms make molecules, molecules, macromolecules, and then the organelles and the cells are this part, this chapter. So, man, I did it again. I shouldn't have messed with it. Shouldn't have messed with it. So the cell is the smallest unit of life. It is to life what an atom is to everything else. And it's going to be something that we'll look at. We're going to break it down fairly simply. Understand they used to call it a simple cell, but now we realize each cell is far more complicated than what we used to think. It is more complicated than an average city. Now, as we look at it, cells, there's a bunch of different types of cells. As it says here, the human body consists of something like 30 trillion cells. That's weird because usually in the same publisher has the 210, 211 book, and it says 75 trillion, and that's a big difference. But trillion is still a big amount, right? It is mind-blowing how many cells are in your body. And cells come in many different shapes and forms, and not worried about that. And we'll look at them as we get into the individual organ systems. But we look at what we call a composite cell. A composite cell, like I said, was this idea of a Franken cell. It's where we take the basic idea of all these cells, these 30 trillion cells, and look at the common parts of them. And in general, a cell is going to consist of a nucleus, the cytoplasm, and the cell membrane. And we said that the cytoplasm is more than just the fluid inside the cell. Remember that cytosol is the fluid. Uh, when we say the fluid, the cytosol plus the organelles, that's what we're talking about with the cytoplasm. Now, the organelles, like we say, are the little organs. That's what it literally means. And so organelles are the organs of the cell. So this is our Franken cell. Again, it's got all kinds of things jammed onto it. Nobody in here should have a cell that looks anything close to this, but it is how we use it to study. So the first thing we look at is the cell membrane. We talked about this being a plasma membrane, and that it is selectively permeable. It has special things on it. Little markers that will tell the body that it's you. Also markers that can communicate with the other cells around it, and so there's a lot of things going on the cell surface. But we're not really going to get into all of those, but just letting you know. It is thin, it is flexible and elastic. It's like a little balloon, but it is what we say selectively permeable. This selectively permeable membrane is selectively permeable because it is made up of a double layer of these phospholipids. And we talked about the phospholipids. This little picture, whoop, I went the wrong way. Nope. That little picture up here, this is showing the double layer. And we'll talk about it a little bit more, how it has the water-loving heads and the water-hating tails, hydrophobic and hydrophilic. And so these lipid-soluble molecules, like oxygen and carbon dioxide, which are the respiratory gases, and steroid hormones, remember, steroid was one of the classifications of lipids we looked at. So lipids were, triglycerides were dietary lipids, phospholipids were another type, and steroids were considered our hormonal lipids. And so things that are lipid soluble can move through this phospholipid membrane like it is not there. Now this is really important when we start talking about diffusion and we're explaining how things move back and forth. But the cell membrane is impermeable to water soluble substances. And this goes back to, again, I hope you remember the idea about the way molecules are made. If it is charged, it cannot get through here. If it has any type of electrical charge, it can't get through. If it is not charged or not polarized, it can. That's the big difference. Now, in this, it talks about cholesterol molecules used to stabilize the membrane. I'm not worried about that. Now, of these proteins that are found in the cell membrane, the only thing I really want you to know is the basic idea of these two terms. Transmembrane proteins means that it crosses the membrane. It has a surface on the inside and the outside. It crosses it. That's why it's called transmembrane. Peripheral proteins are ones that are just on one side or the other. They don't go across. Right? Now, as we look at this, the main thing on here is down at the bottom here where it talks about ion channels. Again, when we talk about that, we're going to be talking about how things move across and diffusion versus facilitated diffusion type things. Now again, cell identification is one of the main things that's going to be on there. I just want you to know that on the surface of every cell in your body, there is something on it that is an identifier. It's going to tell your body that it's you. This is what your immunity works with. The immune cells go through and they're looking for this identifier. If a cell does not have it, it's going to trigger an immune response to it. Not worried about the cytoskeleton in here. And again, not worried about the cellular adhesion molecules. So when we get to the cytoplasm, so going back, the cell membrane, the plasma membrane, the big thing to remember, it is a phospholipid bilayer. It has water-loving heads, which are referred to as hydrophilic. Philly means love. Hydrophilic heads. The outside and inside layer are charged. That's what it means. They have positive and negative poles on them. That's why they love water, because water loves anything that has poles to it. The hydrophobic tails stick in the middle. They are nonpolar. They are fat. So I've got this water-soluble layer on either side and the water-insoluble or fat-soluble in the middle. If something is charged, it will be pulled to the surface, but it can't get across. If something is not charged, it's not necessarily going to get pulled to the surface, but it can move through the membrane like it's not there. So if I increase the amount of something that's a lipid on the outside of the cell through basic diffusion, it will just even out between the outside and the inside. Does that make sense? All right, and that's why it's selectively permeable. So going back now to the cytoplasm. Cytoplasm is the liquid plus the organelles. And so there were some main organelles we looked at. Ribosomes, right off the bat, ribosomes. Remember, ribosomes are the site of protein synthesis. And since last class we were talking about protein synthesis, how DNA goes through a process called transcription and then translation. Translation happens on the ribosome. Translation is protein synthesis. I'm taking that messenger RNA and turning it into a string of amino acids that's going to be a protein. That is protein synthesis. So translation is protein synthesis. All right. Now, they're generally found, ribosomes can be found free in the cytoplasm or in the cytosol, but in general, they're going to be found bound to something called the rough endoplasmic reticulum. An endoplasmic reticulum simply means tubing, an inside tubing. And so rough endoplasmic reticulum is called that because it has all of these little dots on it. It looks rough. The dots are ribosomes. And since ribosomes function in making protein or protein synthesis. Rough endoplasmic reticulum is the same thing, because the endoplasmic reticulum is just the tubing, right? Smooth endoplasmic reticulum doesn't have ribosomes, but there are other cells in there that make lipids. And I told you can kind of remember this because fats are generally smooth. They're slippery, right? And so smooth endoplasmic reticulum is all about producing lipids. All right. Now I'm going to go pretty quick because we're doing the review and trying to get through, but if you have a question, please feel free to ask, right? Now, one of the organelles is just simply called a vesicle. All a vesicle is a membrane-bound sac of something, right? Now, when we make proteins, all right, the rough endoplasmic reticulum, I'm bringing in chapter 4 now since this is kind of a review. So I'm going to have that little transcription that happens in the DNA. Messenger RNA goes out to the ribosome and we go through translation. Transfer RNA goes and gets the individual amino acids and puts them in a chain, and now we have a protein. That is called translation. That protein will then go to the Golgi apparatus. The Golgi apparatus will then modify it. This is where it basically will make sure it has the right shape. We talked about that, the conformation, make sure it has the right shape. This Golgi apparatus does that and then packages it into a vesicle. That vesicle is then sent out to wherever it needs to go, right? It basically is like the post office. It's going to package it, address it, and send it, right? Daniel. Well, not necessarily, because it's inside already, so it'll only be going out. Does that make sense? Yeah. Now it might, if we package a vesicle and it goes out and it goes to another cell and then goes into that cell, that would be kind of endocytosis. Does that make sense? But it's only pushing it, since it's inside the cell, it can only push it outside. Mhm. Would it go into the Golgi apparatus? Not necessarily. I'm not going to say no, I don't know that 100%, but in general, that's not the process. But I wouldn't deny it because I don't know for sure. All right. Good question, though. The mitochondria, again, most people know this. Mitochondria, the powerhouse of the cell. Right. Now, we looked at this, again, cellular respiration. This is used to produce this molecule called ATP. The molecule of ATP is our energy source. And so in cellular respiration, we had 2 phases. The second phase, which includes 2 steps, happens inside the mitochondria. And that second step in there, called the electron transport chain, is where we make a ton of ATP. All right, so just to review, cellular respiration, the first step is called glycolysis. It doesn't require oxygen. It happens in the cytoplasm. It's splitting glucose. If oxygen is present, then the byproduct of glycolysis gets taken into the mitochondria. Now we're in aerobic respiration, which means with oxygen, and it has to have oxygen to get in there. That's where that happens. Right? That's why the mitochondria is called the powerhouse of the cell. Because as long as I can get in here, I'm going to make a lot of ATP. Now, mitochondria have their own DNA. It's a very interesting study on this. And in general, we look at these little, we'll look at the composite cell, and on this composite cell, you'll see like 1 or 2, 3 of them. Your cells in general are going to have thousands of mitochondria in them, right? So the mitochondria, again, powerhouse of the cell, it is producing this crazy molecule called ATP. And if we can get in there, we're going to produce a lot of it. Mitochondria look very weird. Probably not going to get into too much about it, but you will, if you get farther along, you'll study how this weird inner membrane works to help produce it. Lysosomes and Perioxions. Both of these are types of digestive vesicles, just digestive sacs. Every cell is going to have a lysosome in it. Lysosomes are the garbage disposal, as it says somewhere in here, maybe. Garbage disposal of the cell. Its job is to go around and get rid of waste products. And it defends the cell. If something comes in, a bacteria or virus in there, it is one of the things that's going to go and try to take care of it and get rid of it. Perioxiomes, on the other hand, are like lysosomes, but they're very specialized. They're going to be found in areas like the liver. The liver is the main area you find perioxiomes. It's designed on a specific scale. This is going to try to break down anything that comes in, any bad part. This is only for certain things. Again, not going to get into fatty acids and hydrogen peroxide and detoxification of alcohol. Just know perioxiomes are specialized. They're going to be associated with the liver. Excuse me. In here, the cytoskeleton, the only thing I want you to know about the cytoskeleton is it's what gives the cell the framework that it has. There's a lot of things that it does. It's going to, it provides little pathways for things to move on and whatever. But in general, its main job is to provide the framework inside of the cell to give it its shape and structure. Yes. Oh, gotcha. No. I gotcha. It, again, on this only has this one page on it. It could get very in-depth if you wanted it to, and I think that we're probably pretty good with that. Just know it's the framework of the cell. Now, some of the other structures, the centrosome. The centrosome, really, it's weird. It says a non-membranous structure. The centrosome is kind of almost an area. It is a structure, but it's kind of an area because inside the two structures are these things called centrioles. And those are kind of the structures. Does that make sense? So these two centrioles are found in an area called the centrosome. Their job is to wait for the cell to get ready to go to mitosis. When the cell gets ready to go to mitosis, these two centrioles split and they go to different sides of the cell, and they're the ones that pull the cell apart. They're actually the ones that do the main job of cytokinesis. And even in the anaphase, where it's pulling apart the chromosomes, it's the centrioles that are doing the job. All right, does that make sense? But in this, all I'm worried about you knowing is the centrioles are found in an area called the centrosome. They divide and go from the different sides of the cell to help the cell divide. All right. The last two things on here, cilia and flagellum, these are more along the lines of just surface modification. Cilia is just on certain cells, on the surface of it, there'll be little hair-like extensions off of it. That is cilia. And it is simply designed to move things that get on top of the cell. Again, this is like crowd surfers, right? Flagellum. Flagella is only on one type of cell. It's called a sperm cell, and it is just designed to help that cell swim. There's only one per cell, and it is only on sperm. Then we get to the nucleus, the third part of the composite cell. This is where the genetic material is going to be. This is where the chromatin during normal cell life and then chromosomes during mitosis, this is where that's going to be found. It has a very protective nuclear envelope. In the nucleus, this is actually a double phospholipid bilayer. So if we had a phospholipid bilayer, just thinking of it as the cell membrane, in the nucleus, the nuclear envelope is two of them. And in this nuclear envelope, there's no things in it. There's no studded parts to it. There's no communication aspects to it. The only thing that's going to be found in it is little holes called nuclear pores. All right. It is smooth compared to the cell membrane. Now, inside the nucleus, the fluid in there is called nucleoplasm. It is different than the cytosol. Inside the nucleus, there's going to be a darker area called the nucleolus. And in this area, this is where RNA is produced. So when we go in to think that it's time to go through transcription, the RNA is produced here and moves into the genetic area. And this is also where ribosomes are produced. So again, the chromosomes, the chromatin is where all the little recipes are. In the middle of that, I have control to make more kitchens. So if I need to produce more stuff, I'm right there. I produce more ribosomes from that same spot. And then finally, chromatin and chromosomes, which are basically the same thing, just different views of them. Chromatin is loosely coiled, where chromosomes are densely packed. Now, this is where things get a little bit kind of weird when we start talking about how things move back and forth through the selectively permeable membrane. And remember, there's two types of processes in the body. One that doesn't require energy and one that does. It doesn't matter if we're talking about moving across a membrane or breathing or whatever. There are two types of processes. Passive processes happen without the cost of energy. Active processes need energy. And so we have passive transport mechanisms and active transport mechanisms. The passive transport mechanisms, there's 4 of them. Diffusion is simply talking about things that can move through the selectively permeable membrane without any help will diffuse. It is the same as if I had a little balloon and I popped it here and it was full of helium. The helium would eventually spread out through this room. If there was a little wall here and I popped it and the helium couldn't get across, that wall stops it. But if there's no wall, it spreads all the way across. Diffusion is something that can move from high concentration to low concentration because the wall doesn't block it. Make sense? If I have something that I want to get across, but I don't want energy to help, to have to cost energy for it, but it can't get through the wall, I can make special little pores, little things that help it. So let's say it's something, again, glucose. If there's a wall right here and I have glucose and let's say, I'm just going to say butter because everybody knows butter's a fat, right? Let's say I have glucose molecules in butter and I increase the glucose and the butter in here. And there's a wall, the plasma membrane, and there's a little bit of both glucose and butter in here. The butter will move straight across through diffusion. It'll even out because it's a fat. If I have 30 on this side and only 10 here, it's going to end up working where it's going to have 20 and 20. That's diffusion. Now the glucose can't get through here, but let's say I have 50 glucose molecules here and I only have like 10 here and I want to even it out. Diffusion can't happen because it can't get through the wall, but I want glucose to come in. I'll make little holes in it that only fit glucose. And so glucose is going to still diffuse, but I'm going to help it. So we call that facilitated diffusion. Facilitated means help, make happen. All right, does that make sense? And it'll end up being where if I had 50 on this side and 10 on this side and it can get across, now we're going to have 30 and 30 because it's going to even out. All right. Jumping over osmosis, we go to filtration. So I got this little selectively permeable membrane and I can't necessarily get across. I don't have special carrier molecules and stuff to do it, but if I increase the pressure on this side, I can push it across. Now it doesn't cost energy, but it's going to cost pressure. And again, when we get into the circulatory system, you're going to see as those capillary beds push things out, there's a giant pressure drop as the blood goes through the capillary system. And that's used to push the plasma out carrying the oxygen. All right? So filtration doesn't cost energy, but it requires some pressure behind the substance. And then we get to osmosis. Remember, osmosis is all about water. first thing you should think of in this as you're learning it is whenever you hear osmosis in your brain, you should automatically just think water. Osmosis is the movement of water. So again, I have this little membrane right here, and I have salt water on this side, and there's a ton of salt in this part. And then on this side of the membrane, I've got a little bit of salt. This has got 1 tablespoon of salt on this side, and this has, 4 cups of salt. Right? So salty, concentrated, dilute. We'll say the salt can't cross the membrane here, so what happens is water wants to make it even. So again, just think of it. If I have a cup of water that has a little bit of salt in it and a cup of water that has a ton of salt in it, if I want to make them even, I'm going to take the water out of the dilute part and put it into the concentrated part. Right? I'm watering down the one side. So if this was the case and this side had a bunch of salt in it and this side only had a little, the water would move from the area that was dilute to the area concentrated. And it's trying to dilute this part, try to water it down. Right? Make sense? So water's the thing that moves, not the solutes. And water will always move from an area of low concentration to high concentration because it's the water trying to even out the concentration. Right? And so the thing for us is when we're looking at how this would affect in the healthcare, I mean, if you put a solution into somebody's body, it can cause problems if it's not the right osmotic concentration. And so we look at 3 different ways this can affect body tissue, and we look at it by putting a blood cell into a solution. If I put a blood cell into the solution, I've got water, the cytoplasm inside the cell. It's got a certain concentration. If I put it into a solution that has the same concentration as it, we call it isotonic. What it's saying is I've got this membrane and I've got the same concentration here and the same concentration here. So nothing's going to happen. Some water will enter the cell, some water will leave. It's always at equilibrium. Yeah. It does, but it's still there's still water movement, but because it's the same concentration on either side, once water enters here, another water molecule goes back that way because it's even. No, it's diffusion is about the solutes. Yeah, the diffusion's about the particles in it. And since the particles can't move here, but the water can, the water's trying to even out the concentration. Now, if I take it, I'll go to this one first, and I put it into a solution and I see that the cell starts shrinking, I know that means that water is being pulled out of the cell, right? Don't worry about the name of it, whatever. But if I'm saying, all right, water's being pulled out of the cell, and I know that water moves from dilute to concentrated, all right, that's telling me that the cell is dilute versus the solution it's in being concentrated because water is moving out of the cell. Make sense? As long as I remember, water moves from dilute to concentrated. All right. So this is called a hypertonic solution. And I said the easy way to remember it is the E is a little shrunken cell. It's an O that's kind of shrunk in on itself. And so you can remember a hypertonic solution is when the cell shrinks. And again, if the cell shrinks, that means water's leaving. As long as we know water goes from dilute to concentrated, it's telling me that the solution it is concentrated. It is pulling water out. If, on the other hand, I take and I put the cell into a solution and all of a sudden the cell starts swelling up like this big balloon, that's telling me that water is being pulled into the cell. And so if I know that water moves from dilute to concentrated, this is telling me that this cell has been put into a dilute solution. That is hypotonic. And again, you can remember the O is like this big swollen balloon. After it, that's what the cells will do. they can only accept so much, and then they'll start rupturing. Yeah. That would be an example of necrosis. Yeah. All right. Now the active transport, or the active mechanisms, the main one is active transport. So let's say, again, we go through this, we got a little wall here, I've got a ton of stuff over here, and that's the outside of the cell, and I've got a little bit here. We said diffusion is if the stuff here can move through, it will balance out. If we have facilitated diffusion, what we're saying is stuff here that can't get through the membrane, if I have a little hole that it can get through, it will diffuse. It's just facilitated. We're helping it. But if I have a membrane here, and let's say I only have a little bit of something here, this is outside the cell, and on the inside I got a whole bunch of it. Even if it could move through the cell membrane, it's not going to move into the cell because it's going to be moving against the concentration gradient. Right? Because we're talking back about solutes now. And so if I want to get something to move from a low concentration to a high concentration, a solute, I have to have a pump. I have to have something that's pulling it. It's literally pushing a boulder uphill. I'm going against the gradient. All right. So again, if I have, so what we have on this side, 6 students here, and let's say that all the chairs were filled in here, so 24. So we got 6 on this side, 24 on this side. If we were talking about diffusion, what would happen is you on this side would end up moving over so it would balance it out, right? If this membrane is separating it, but I want y'all to move to this side, that's not going to happen naturally because of the concentration. Does that make sense? So what I'm going to end up doing is grabbing people and pulling them across. That's called active transport. It's a pump. I am moving something against what it wants to do. What the natural instinct would be is I want to balance it out. I'm moving it against that natural instinct. So active transport is moving things against the concentration gradient. You're not pointing, Daniel. Okay. I wonder if the secondary active transports, which we're not going to get into, will have you can have a hydrogen or a sodium it'll use to pump things across. Right. So in this, this is showing when we get into the nervous system, when we see how axons send signals, there's a sodium-potassium pump that helps regulate and set up the concentration differences, and it's against the gradient. I'm going to have a lot of sodium out here on the outside, a little bit of sodium on the inside, comparatively speaking, but I need to get the sodium to get back out. It won't do it under normal conditions because it's concentrated on the outside. So I got to pump it. And so it costs ATP. So the big thing about this, active transport is about moving things against the concentration gradient, moving solutes against the concentration gradient. And because of that, I have to have pumps to make that happen. And it's going to require energy. And so this is an active process, and we call it active transport. Now, the other type is the energy and the cost is going to be of the cell membrane. And we call this endo and exocytosis, coming in or leaving the cell. And a part of the cell membrane is going to be used to create a vesicle and pinch off, either coming in or coming out. God, my nose is itching. I'm sorry. This shield goes against what I naturally. Anyway, sorry. So, endocytosis is moving things into the cell. Exocytosis is moving things out of the cell. Now, in endocytosis, there are a couple of types. One is called pinocytosis. This is when it is liquids coming in. This literally means cell drinking. The other one is called phagocytosis. This means cell eating. This is when it's a solid particle. This term you're going to hear many, many times from now on, because there are lots of cells that are considered phagocytic cells. They engulf other particles and digest it. Vesicles work by phagocytosis. They engulf particles, digest it, right? And so these are both types of endocytosis. It's just, trying to be specific between liquid and solid. And then the other one is called receptor-mediated. Now, again, we're not getting really far in depth in it. Understand that this is going to require receptors. I mean, I think that's pretty much a duh type thing, right? Receptor-mediated endocytosis requires receptors. And what this is saying is I've got a cell membrane here, and I've got things on the outside that I don't always want to have come in. When I want them to come in, I'll create a receptor, put it on the cell surface, it'll bind to it, bring them in. When I don't want it, I quit making receptors, right? So the receptors mediate it. So those are how we move back and forth. Passive processes are diffusion, where things that can move across the cell membrane are just going to move according to the concentration gradient. Facilitated diffusions are things that would do that, but they can't get across the cell membrane, so we produce little pores that are specific for them to make them do the simple diffusion. Filtrations, when we just shove them through because we have pressure behind it. And then finally, osmosis, which is all about water. It's when the solutes can't move, right? The little particles we've just been talking about with diffusion, they can't move. So water will move in order to try and make the concentration even. Now, when water moves, it moves what would we consider opposite. It moves from a dilute area to concentrated area. But if you can think about that, it makes sense, because what it's trying to do is make the two concentrations even. And the way to do it is take water out of the dilute part and put that water into the concentrated part. That ends up making the dilute part more concentrated and making the concentrated part more dilute. All right? And so osmosis is all about the movement of water. When we talk about the active transport, the type that are the active processes that need energy, active transport's the first one. And this is actually going against the concentration gradient, where I'm forcing molecules to do something they normally wouldn't. I'm pushing it uphill, so it costs energy. The other one, the other one is endocytosis, where we've got two phases. It's either liquid or solid. Pinocytosis is endocytosis for liquid. Phagocytosis is when it's solid. The other type is receptor mediated, and that's just saying that I need receptors to make it happen. All right. The cell cycle. How's everybody feel about the cell cycle? Okay. Before I move on, are we okay? I mean, and you're free to say it's okay. There's nothing wrong with saying, man, I'm lost. It's okay. Are we okay? All right. The cell cycle. All right, remember that the entire cell life is considered in the cell cycle. Right? It has interphase and then mitosis and cytokinesis, which both are when the cell is splitting. Mitosis is all about splitting the genetic material. Cytokinesis is when everything else is splitting. And so interphase is kind of like normal cell life. Now, it says here cells do not usually continually divide. So they have these countdown clocks we call telomeres. Every time a chromosome is split, a little part of that gets knocked off, and it can only go so far. Once the telomere is gone, that's when that cell will no longer be able to divide, and so it just normally dies. It reaches the end of its life, and boom, apoptosis. All right. Now, for a cell, now understand, and this is kind of weird, none of these cells are created, really. It's just the cell divides. Each one, like there's two daughter cells here. While we can consider them created because they just got split apart, all they really are is the original cell divided. Right? So it's not really like it created. So it's this weird cycle. So let's take one of these daughter cells and say it just split. We just got it fine, get the division here. It starts into what we call interphase. Interphase is normal cell life. G1S, G2. I'm not worried about G1 and G2 as far as things that I want you to pick on. I do want you to know that the S phase, this middle phase, is where the DNA makes a copy of itself. You can kind of say it splits, right? It splits apart, makes two complete copies. That's so that once we get back here, these two daughter cells now each have a copy, right? So DNA replication happens in the S phase. Now, once interphase gets through here, it's going to get to a point where all kinds of signals will be sent out, and the cell will know, all right, now it's time to start going through division. The division is mitosis. And again, mitosis is all about the dividing of the genetic material. The four stages of mitosis are prophase, metaphase, anaphase, telophase. And I said that if you kind of include interphase, you can say, I passed my anatomy test. So pass my anatomy test, or PMAT, is mitosis. Prophase and telophase are more processes, where metaphase is a quick snapshot and anaphase is just a little bit of a longer than snapshot picture. But the prophase and telophase are the two long phases in this. And so in prophase, right, prophase is where we're going to see a lot of things happen. And again, like I said, if you're just sitting there and have time on your hand and a really good microscope and thought, I'll just sit there and watch a couple of cells, it might get to be where you're watching it and all of a sudden, instead of this just dark, uniformly colored kind of dark thing with one little dark spot in the middle, which is the nucleolus, all of a sudden you'll see it start, it almost looks like it's clotting. It almost looks like it's like milk, you know, it gets, all of a sudden it'll start changing where there's a lot of little dark parts. And then right then, you know, this cell's starting to go through mitosis. The wall around it will disappear. You can see there's no boundary between the nucleus and the cytoplasm like there is here. So the nuclear envelope will disappear. You'll start seeing things get from one side to the other. It doesn't really show it here, but that's a process. And as these now what we call chromosomes start lining up in the middle, you'll see them start migrating, and then all of a sudden they'll get in the middle. And at that point, you know you're in metaphase. Metaphase, you can easily say this is where they meet in the middle. I'm met in the middle, metaphase. It's not going to stay there really long. It's like a weird football game. They get all the line of scrimmage, and instead of hut and going at each other, they say hut and run away from each other. So they line up, boom, get pulled, they break apart. Now we're in anaphase. The chromosomes have split. They're getting pulled from side to side. At this point, cytokinesis starts, because it's not just the chromosomes that are getting pulled from side to side. Now everything is getting pulled to side. But really quickly, this starts reorganizing. You'll start seeing these very distinct chromosomes starting to unwind and not be these really distinct chromosomes anymore. You'll start seeing a dividing line, which we call the nuclear envelope, starting to form, and the cells starting to split apart. At that point, we are in telophase. Now, if I'm asking you this, it's not going to be pictures. It's going to be things I'm talking about. Metaphase, without a doubt, you should know, meet in the middle. Anaphase, I would say this is when things start to divide. It's getting pulled from side to side. Prophase, going back to the beginning, I would say, look, the chromatin's starting to coalesce into chromosomes. The nuclear envelope starting to disappear. The chromosomes are starting to line up towards the middle. If I'm talking about telophase, the end phase, I'm going to say the chromosomes are starting to unwind. The nuclear envelope is starting to form. The cell is getting ready to pinch into two. Right, you see the difference in all of those? It'll be matching, right, or multiple choice, some way or shape or form. But there's certain key things for each one that are indistinguishable. I don't know. I can't speak today, but I think you know what I'm talking about. Maybe. All right. Now, outside of mitosis, there is another type of cell division that is strictly for reproductive cells. I call it meiosis, right? This is simply meant to divide these cells to have half the genetic material. We're not going to get into the different parts of this and what all it means, but you do need to know meiosis is about producing reproductive cells. Reproductive cells they cannot have the full genetic material. If we have 46 chromosomes, if the sperm has 46 chromosomes and the egg has 46 chromosomes, that's a problem. Does that make sense? So meiosis is making very special cells. They have half the genetic material, so that when they get together, they got the full deal. Now, cytokinesis, again, this happens in anaphase. Cytokinesis, the only thing I want you to know about cytokinesis is this is the division of the cytoplasm. This is everything else except the DNA. So if I went back to this little slide, oh, where's the little cycle? This. And I was going to compare these two cells. These two cells that we just created would have identical DNA, but they would have different cytoplasm. They would have different amounts of ribosomes and different amounts of mitochondria between the two, because the splitting of everything else, the splitting of the cytoplasm, isn't even. Does that make sense? So the two daughter cells are going to have the same DNA, but different everything else. This part right here, and again, I'm not going to test you on it, G1 is all about each one of these cells going through inventory. Did I get enough receptors on this? Do I need more glucose channels? Do I need more ribosomes? Do I need, whatever? Or do I have too many of this? That's what this phase is. And it's because the two cells aren't even on the other things. All right. Now, as the cells grow, they will become specialized. Now, when we sit and look at the cells, remember, we start with an egg and a sperm getting together. That first cell, as it starts dividing, there's no difference in the original few cells. You can't tell what any of them are. You couldn't tell the difference between them no matter how you looked. But then at a certain point, the cells will start changing and you'll start being able to tell different ones. At this point, you'd be able to tell these cells are going to end up being the skeletal system. These cells are going to be the nervous system. These cells are going to be the GI tract. We call that differentiation. They become specialized. All right, they follow their distinct path. Now, certain cells will remain in certain areas, and we call them stem cells. Stem cells have the ability to produce many types of other cells in their particular field. For instance, we have a red blood or a blood stem cell that hangs out in our red bone marrow. If I have an infection and I need one of the 5 white blood cells, my body's going to send out a hormone. That cell will turn into a monocyte, for instance. But that same stem cell, if I donate blood and my body notices I'm low on red blood cells, a different hormone will go to that same cell and it'll make red blood cells. If I am having problems with my blood clotting, that same stem cell can turn into platelets. Right? So all a stem cell is something that can turn into multiple different types of cells. Not worried about. Yeah, but there'll be another stem cell that'll take it. It kind of divides and hangs out one stem cell, and then the other ones follow through. Yep. And so if anybody's ever, hopefully no one in this class has ever had to deal with needing a bone marrow transplant, but that's what they're harvesting. They're actually trying to get the blood stem cells. All right. And then again, here we go with cell death. Cells have a life cycle. Now, as long as they're going through mitosis, they really don't die. They just keep making new cells. But like we talked about, each time it divides, there's a little part of that chromosome that gets taken off. We call it a telomere. And once it gets to where there's no more of that, the cell will not go through mitosis anymore, and it will just die. And it's normal, and it's called apoptosis. However, I don't understand why this doesn't really talk about necrosis, but its opposite is if the cell's damaged, it dies, and we call that necrosis. Necrosis is abnormal cell death. All right. So chapter 3 went through composite cell, talked about the nucleus, the cytoplasm and their organelles, and the cell membrane. Talked a little bit about how things move across the cell membrane, and then how cells multiply by mitosis. All feel okay? All right. Okay. No, you're fine. Look, like I said, I'd rather that than people that just stare at me like we're playing poker or something and they're trying to fake me out. I want to see if you're getting it or not. So chapter 4, and I get both of them. Again, I understand. I don't want to sound like if you're just not just staring at me that I think you're a bad person or something. I just don't. I want to know what's going on in your head. Um, well, as far as this goes. So chapter 4, and again, now I've got the PowerPoint videos for that, so that'll probably be a lot better than this, but just to do the little review, we're talking about chapter 4 is about cellular metabolism, the chemical processes that go on in each cell. And so in this, we really concentrate on these amazing proteins called enzymes. Enzymes, again, are chemicals that are produced by our cell that will cause chemical reactions to happen. And so it is what controls metabolism. So cellular metabolism, as it says here, is a group of chemical reactions that acquire, store, and release energy in the cells of the body. Energy is derived from chemical bonds and nutrient molecules obtained from the diet. So one of the big things about something like glucose is it, C6H12O6. So for every one water molecule, H2O, we've got a carbon on it. And the way it's laid out, the carbon-hydrogen bonds are energy bonds. And so that's what we're doing is as we break down the hydrogens off of the carbon, it releases energy. It ends up causing us to be able to use it. So it's chemical bonds that our body uses to put together or break apart to store or release energy. So anabolic reaction, where we're taking smaller molecules and building larger molecules out of them, I'm making bonds. This is storing energy, right? It's costing energy, requires energy. Catabolic reactions, where I'm breaking things down, release energy, because when the bond breaks, energy is released. Now, in any of these reactions, as we're breaking it down, most of the energy is going to be released as heat. That's why we have a higher than normal body temperature. Our body temperature, most of us probably have houses that the thermostat set at least in the mid 70s. I don't know. I mean, I like it really cold. Some people don't, but I'd say at least 75, 76 is probably what. Yeah, I'm with you. I like it cold, but at a 98.6 body temperature, our body temperature is about 20 degrees above what our normal surroundings are, and it's all because of this release of energy that our body is producing in all the chemical processes. Now, remember anabolism. We talked about anabolism. The building of things is made by dehydration synthesis. Synthesis means to build. So dehydration synthesis literally means to build by removing water. Catabolic reactions are made by hydrolysis. I break it down by putting water in the middle of it, right? So water's still the key. Now, that's where this was. I thought we talked about it at a different time, but I guess I didn't. So this is where I talked about it. So dehydration synthesis, again, I don't like the way they put this there. I think they could have drawn it a little better, but that's just me, and I'm not going to draw on this since we got the little sheet and they don't like me doing that. But all we do is if I am making a larger molecule, I take A plus B equals, makes it AB. I'm taking a hydroxyl off of one side and a hydrogen off of other and producing water, and where that bond was broken, a new bond forms. and we make a larger molecule, dehydration synthesis. When we make when we break something down, it is hydrolysis. If I have a molecule that is bound together, I take hydrogen, I jam it into this spot, it breaks it down. A hydroxyl's on one side, a hydrogen's on the other. I've made a large molecule smaller by breaking it apart with water. Now, the way this works is water just can't get in the middle of it, and water can't always just be forced to pull out of it. The way we talked about it is saying that, if we're all molecules and we're moving around and we bump into each other because we're blind, it's not necessarily going to cause a chemical reaction. For molecules to bind together, they need to do more than just bump into each other. It has to be where they hit just right, right? And so the odds of, any two people in here hitting just right is pretty low. And so what our body does is it controls those reactions by making these things called enzymes. Enzymes have a special seat in them that is only able to sit two different molecules, these two things called substrates. Nothing else can sit there. And when those two molecules sit there, they are forced to sit in the proper alignment to make the bond happen. And enzymes can either be used for anabolism to build things up or catabolism. Either way, what they're doing is they force the molecules to be in a certain specific orientation so the bond will form or will break, either way. But without the enzymes, this reaction, any of the reactions we're talking about, would not happen. All right. So these are what make us live. Yes. They will attract. They actually attract. They go and scoop it up. Yep. They go on the hunt. All right. And so that's what it means when it says activation energy. Because what would happen is if you ever seen like old like baseball video like Babe Ruth and and it's always faster than normal, right? So imagine that we're normal in here moving around and blind. The more energy we would put in, the faster we would move. In order to make a certain reaction that would happen that would normally not happen, I'd have to put a ton of energy. We would be like these little fast little things moving around. That's what we refer to as activation energy, the energy needed to make it happen. And so if I put an enzyme in there, I don't have to speed everything up because the enzyme's going to make it happen. So that's what it means when it says it lowers the activation energy. Now, the type of enzyme, again, enzymes are specific. The type of molecule that it is specific for is called its substrate. The substrate, yeah. Just remember that whatever the enzyme is designed to work on is called its substrate. Nothing else can sit on it. Nothing else can be used by it except that substrate. And on the enzyme, the little particular binding site area is called the active site. And so on this picture here, it shows the active site. Know that enzymes are not changed by the reaction, so they're reusable. And then when we don't want the reaction to happen anymore, our body will produce something to break this down to stop it from happening. All right. So this is how our body controls the chemical reactions in our body. All the things that are going on, and there are hundreds of thousands of things going on right now in your body. This is what's controlling them, making them happen, and then when we don't want it, we pull the enzyme away and it stops it from happening. Now, most chemical reactions are what we refer to as a pathway, and so enzymes generally control it. Now, the way this works is the very first enzyme is generally called the rate-limiting enzyme. If this is a box of Legos over here and there's a whole bunch of different things and it could end up being different products, our body keeps them in store, and then when we want a certain product, it's going to produce an enzyme that's going to go and get the right Legos, put them together. Once those Legos are formed, then there's a chain reaction that ends up with the product. This is called the rate-limiting enzyme. As soon as this is produced, it's going to happen. When they're not producing it, this reaction isn't going to happen. And so my body can control what we have over there by producing this one enzyme. If I produce it and I'm going to make enough of this product. I've got enough. I don't need any more. My body quits making this. This product, this whole assembly line shuts down. I use the product till I need more. I produce this. I start making it. I got enough. I quit making it. That's negative feedback, by the way. I'm making it till I got enough. I don't. Cut it down. I bring it back. All right. And again, not trying to be speed reader, but just trying to get through. Now, enzymes have things that help them do their job. Cofactors and coenzymes. Remember, coenzymes are vitamins. Again, I usually try to say, the Z and the V are two letters that don't get used a lot. That helped me when I was in school. Remember, coenzymes were vitamins. Cofactors are elements. They're really, again, in dietary terms, they are minerals, right? And so they would be things like calcium and sodium, and zinc is one, again, that is easy to see that zinc's an important mineral for your immunity, and it's because it's a cofactor. If the enzymes, because there are proteins, if something happens to their shape, we call that they are denatured, denaturization. Denatured means that it's changed its shape. If I've changed the shape, that means it cannot do its job. The active site is no longer able to accommodate the molecules. It won't work. That process shuts down, and if that happens in a bunch of our things, we die, right? There are plenty of things that can denature a protein, but the one thing that we don't have control over is the pH. Our body is constantly changing our pH. We have to go through a wrestling match, no matter how clean we live, how perfect we eat, exercise, whatever. pH is always an internal battle. The other ones we can kind of avoid. Please don't stick your finger in light sockets. Don't try to drink things that have skull and crossbones on it. We can usually use our common sense for a lot of the other things. But common sense is kind of the least common thing around. So anyway. Energy, which is what this is all about, making energy. Energy is the ability to do work, and chemical energy is what our body uses. Now, cellular respiration is what we're looking at when we're talking about making energy. I'm not worried about oxidation. If you get into, farther along in biochem, you'll learn about oxidation reduction reactions. And here, I just want you to know cellular respiration is how we're making ATP. Right? ATP is the molecule of energy. You should know that ATP means that there are 3 phosphates, triphosphate. Its more stable cousin is diphosphate. This is how our body keeps it. There's a little cycle. We bind this 3rd phosphate on as a high energy bond. When it breaks down, it releases energy, and we go through this cycle. Now, cellular respiration, remember, is a 3-step process, and there are 2 phases. The first phase is anaerobic because it happens without oxygen. The other phase, the second phase, is called aerobic because it needs oxygen. The first phase only has 1 step, the very first step, glycolysis. Glycolysis happens when glucose enters the cell and gets broken down. When it breaks down, the byproduct, if oxygen is present, gets taken into the mitochondria. If oxygen is not present, we said it makes what we call lactic acid. That's when you get sore, you over-exercise. You no longer were able to use oxygen efficiently. And so after glycolysis, if we have the ability to use oxygen, we get into what we say is the aerobic phase. This happens inside the mitochondria. This step, the first step in the mitochondria, which is the second step overall, is called the citric acid cycle. After that, we go into what's called the electron transport chain. The electron transport chain is where we hit the jackpot for ATP. We make a ton of ATP in this. And so, again, 3 steps: glycolysis, citric acid cycle, electron transport chain. 2 phases: anaerobic, which is only glycolysis; aerobic, which is the other 2. The first step, glycolysis, the anaerobic phase, happens in the cytoplasm. The other 2, citric acid and the electron transport chain, which are the aerobic respiration, happen in the mitochondria. That is why the mitochondria is called the powerhouse of the cell. I'm not interested in knowing the specific number, but you should know that it is around 34, right? 34, 38, you should know it's the mid 30s if I can use oxygen efficiently. Now, I'm not worried about the specific ones because different books will have different numbers, but you should know it's in those mid, those low to mid 30s. If I ask you how much it produces and I give you options of 4, 18, 30, 34, and 100, you should be able to pick the one that's right on that. With me on that? Like, I'm not going to put it and make it all real close. But you should have an idea. All right. And then we got to the whole process of protein synthesis. All right. Now we're kind of running out of time. Protein synthesis we looked at this last Tuesday. So hopefully that's a little fresh in your mind. But remember, 2 phases or 2 steps. In this one, the first step is called transcription. It happens inside the DNA. Messenger RNA makes a copy. Messenger RNA leaves. The second step happens in the cytoplasm on a ribosome, and it is called translation. That uses transfer RNA to look at these 3 codons, make an anticodon, get the appropriate amino acid, and bring it back into place. All right. Remember, the last thing is, remember that if I have a group of letters and say, all right, I want you to tell me what the complementary pair on the DNA is, it's as simple as knowing that anywhere there's an A, there's a T on the other side, or if there's a T, there's an A. They go together. C and G go together. If I ask you I give you a strand of DNA and say, I want you to tell me the complementary pair of the messenger RNA, it will end up being the same as the DNA, except anywhere there's a T, there'd be a U. Only change. All right. People try to make a big thing. If there's a T over here, no, it doesn't matter because the A is there. The only change is if wherever there's an A, there's a U. Over here, wherever there's an A, there's a T. All right. All right. I think I got it.